So I'm going to talk about zero valent iron in a way that might be a little bit different than what you would expect, but it's a talk in two parts, and the two parts were selected to appeal to a fairly broad audience of students and faculty who some of whom are probably working on ZVI, but some of you are not working on ZVI. And so I tried to make the talk have some breadth so that it would be meaningful to all of you. I want to start at the beginning by acknowledging some of the people that have done much of the work that's come out of my group in this area over the years. And many of those people have been Chinese graduate students or postdocs or visiting professors. And so some of them are shown in these photographs here. I'm not going to dwell on naming the names and the faces, but I would just mention that these four will show up in some of the publications that I'll be talking about later on. Okay, so we're going to go back to the very beginning of ZVI. So the very beginning of ZVI was almost exactly 25 years ago. And the definition of the beginning is really this symposium that took place at a national meeting of the American Chemical Society in April 1995. It was in Anaheim, California, and I was the organizer together with Martine Reinhardt, who's professor at Stanford. And this was the first meeting when all the people that were involved in researching this topic at the very beginning, they all came together at this time for the first time in one room, and it was very exciting. It was scary. It was intense because everybody was in competition to be the first to get a publication on a topic. It was extremely interesting time. If you have a big computer screen, you can probably read the names on the table of contents here. However, most of those names you won't know. Probably the only two that you'll recognize are me, that's here and a couple other places, and Renee Schwarzenbach here. But all the others are no longer really working in the field anymore. I'm essentially the last of this first group of people that's still active in the area. But this conference led to a booklet of extended abstracts. At the time, this ACS Division of Environmental Chemistry used extended abstracts. So they were 3, 4, 5 pages long. They're not peer reviewed, but they were quasi publications. And they tended to contain data that was pre-publication in a journal. So if you can get access to these old books of extended abstracts, you can learn about what people were doing before they published their papers. And so I had access to all these extended abstracts, and I put them together in a booklet, which is what you see the cover in the table of contents here. And I recently have made that available online. So if you're interested in this field, or if you're interested in the history of environmental engineering, you can go to the Zenodo website, Zenodo. It's an open access repository based in Switzerland, and you can download the full PDF of this collection of extended abstracts. If it's very interesting, you can search the document for keywords for topics that you might be working on now. You might assume that they are new, and I predict that most of you will find that somebody was already interested in or working on that topic in 1995. For example, at the end of the talk, I'll be talking about sulfidation, so the combination of reduced sulfur and zero valent iron. There are three papers in this list here that are about sulfur and ZVI back in 1995. So I recommend that you download this and look at it. It's both fun and interesting and informative. So back at the time, early on, there were a number of things going on. One of them was trying to understand what happened between the zero valent iron and the contaminants that were adsorbing and reacting from solutions. So in this cartoon here that I is a color version of a figure that I published in 1998. This RX is the halogenated hydrocarbon contaminant. It absorbs to the surface and it reacts at the interface where the iron is underneath and the oxides on the surface. And we were all interested in exactly what is happening at this interfacial region. So that was a lot of what people were working on. On the engineering side of things, most of the emphasis was on permeable reactive barriers. So those are used for remediation of groundwater. And that was really what people were doing back in 1995. Since then, the field has grown in multiple directions. And this Venn diagram I updated several times in the years. This current version includes nanoparticles, which was a big new phase that took off from the original ZVI work, and then the sulfidated versions of the zero valent iron, which I'll talk about at the end of the talk. There's another circle here, which is for ex situ reactive filters. So here, this is groundwater, this is subsurface, this is anaerobic and reducing conditions. And up here, these are ex situ aerobic oxidative processes, and that's also become an aspect of this field. So that all of that interest has led to a lot of papers, as you undoubtedly know. And 5 years ago, I made a graph where I extracted a bunch of data from the literature on the bibliometrics of the zero valent iron field. So this is data on numbers of papers and numbers of citations. And in this figure here, I plotted the numbers of citations, per paper on the y-axis, and I plotted them on an x-axis, which is year of publication. So down here, you see that there are many papers that have a modest number of citations, or on the order of 50 to 100. I would point out that that's actually quite good compared to other fields. 50 citations for the median or citation rate is actually quite good. So this is a healthy area of research. But most of you are looking at the ones that I've labeled up here that have the really high citation rates. There are two main things to see here. Number one, it pays to be first. So these two papers here are the very first significant papers on ZVI. Bob Gillum's with Stephanie Johannesson and mine with Leah Matheson. And they have by far the highest number of citations. In my second paper on ZVI is here, and my third paper on ZVI is here. And they also have high citation numbers. This paper here is the first paper on nano ZVI. And again, it gets very high number of citations. And this is the second paper on nano ZVI. And this is the third. And this is the fourth and fifth. So you can see that nano ZVI drove high citations in this area for quite a long time. Notice, as I said, that this figure was only created in 5 years ago. So the numbers run out here. So you might wonder, well, what's happened since then? Well, I can tell you this. Somebody pointed out to me a few days ago that my first NZVI paper, this one here, just passed 1,000 citations. So if you look over here, it was 500 citations at the time that I made this figure. So it's doubled in 5 years. And I know that this paper here has close to 1,800 citations now. So it also has doubled. All these other papers won't have gone up that fast, but that's an indication that the field is continuing to grow. This version of the figure here shows two slightly different things. It shows numbers of papers per year, which is, as you can see, is growing steadily. And again, it just cuts off because the data are old at this point. It's going up. And this in the green here is total citations per year. And as you can see, there's a huge number. If you integrate under this curve and this curve, you get these grand totals of 4,000 plus papers and approaching 100,000 citations as of 2015. So the field has obviously been very successful. So you might wonder why, because in the overall scheme of things, remediation with zero valent iron isn't the most important thing out there, right? I mean, climate change and COVID and a lot of other things are really more important than this. Why does this field get such high citation rates? Well, a few years ago, I had an idea to explain that, and I wrote it into a book chapter, which is shown here. So the chapter is in a book on aquatic redox chemistry, which I edited it came out in 2011. The chapter was primarily about organic coatings on ZVI, but the theme in the first third of the chapter was the issue that I'm alluding to here, which is the idea that zero valent iron, sorry, I'm having trouble with the mouse, zero valent iron forms the basis for model systems that have been used in fundamental and applied studies of a wide variety of environmental processes. So model systems is the key word. That's the theme that I'm going to emphasize for the next few slides. I want to try to convince you that zero valent iron has become one of, if not the most important model system in environmental engineering. Now, the idea here is that many of the best ZVI studies are not primarily motivated by cleaning up contamination, or they're not primarily motivated by zero valent iron. The authors are not really concerned with that. They're concerned with some bigger, more fundamental, more high level general issue, and they're just using zero valent iron to study it. So I've listed 8 examples here. I don't have time to talk about all of them. In blue, I've listed some of the authors that I think wrote or did some of the seminal early work that best exemplifies my point here. But I'm not going to have time to talk about most of them. The only 3 I'm going to talk about are the ones that are highlighted in red: dechlorination pathways, number 2, example 5, kinetic model of surface reactions, and number 8, graphical methods for data analysis. So let's look at the first one. That's example number 2, the dechlorination pathways. So for many years, huge numbers of research projects and field studies were done on the environmental biodegradation of chlorinated solvents. So pick trichloroethylene, this structure here, as the prototypical chlorinated solvent. And what universally would happen would be that this compound would undergo reductive dechlorination by hydrogenolysis to remove one chlorine, replace it with a hydrogen, remove another chlorine, replace it with a hydrogen, remove another chlorine, replace it with a hydrogen. So you sequentially go step by step through the dechlorination steps. That's called sequential hydrogenolysis. The problem with that is that this pathway is fairly fast, but this step is much slower, and this step is even yet quite a lot slower. So in almost all cases, what would happen is the parent contaminant would go away, and you would get accumulation of these intermediates because they didn't degrade very fast. And so they have been often referred to as stall intermediates. So stall, the word stall in this case means like if you stall your car and it won't go, right? So it's the same idea here. These intermediates stall, the degradation stalls at this point. And these are toxic and they're hazardous, and they were a big problem, a big, big problem. Around 2000, Lynn Roberts' group at Johns Hopkins University was studying zero valent iron. And they realized that degradation of TCE with zero valent iron was mostly not going this way. It was mostly going this way. And these arrows, these diagonal arrows here, are for reductive elimination. And that's a different pathway. It's a different mechanism. And the most important thing is these intermediates don't get stuck. They don't create a stall. They don't accumulate in the environment. And that's very desirable because it avoids the problems that they had here. And the engineering community has become very interested in this, and they've now started referring to this as the biotic pathway and this as the abiotic pathway. That's a dangerous oversimplification, but it's also mostly true. And so this is my overall point here is that this was discovered by people that were studying zero valent iron. Before that, when everybody was studying biodegradation of chlorinated solvents, they didn't understand this dynamic. And just to illustrate my point that this has been tremendously influential to the field of environmental engineering, I did a quick Google search for a practical example of this, and up popped a new page on the website of this company, Serum Lab. Serum Lab provides services for people that are cleaning up contaminated sites. They provide advanced analytical analyses of field samples. And we don't care about the details here. The point is just that they were working with a group in Denmark that was characterizing this site that had a great big plume of chlorinated solvents. They did a detailed study to characterize what was going on in the degradation pathway using advanced molecular methods. And they concluded that the big red arrows are hydrogenolysis, biotic, hydrogenolysis, biotic, reductive elimination is abiotic. And this branch here is what they all want to do that, because that's better than creating these recalcitrant intermediates. So it's a big deal. And it was discovered with ZVI. So another example, this is number 5. And this comes from my group. This is simply the formulation of how you describe the kinetics of a surface reaction in an aqueous medium. And this came from our 1996 paper on the kinetics of contaminant degradation by ZVI. And we wrote these rate laws, carefully assigned symbols and markers for these two expressions here. And we provided a little bit of data to try to justify that this was the right model. In years since then, this formulation has become almost universal. You see people talking about KSA in many, many, many papers. The first place that you will ever find it is in this 1996 paper that we did on ZVI. So now KSA is even used in other fields when they're not talking about ZVI and they're not even talking about contaminants, but it came from this paper. So again, this emphasizes my theme about how ZVI really was a model system for developing a general kinetic model for interfacial reactions. In subsequent years, we expanded that a little bit. We took the standard model from the previous slide. We rearranged it like this here. And now we can say, okay, if we equate this term here with this term here, then we get Km equals KSA times A sub S, which is the specific surface area of the particles. That's this expression here. If we take the log of this expression here, we get this expression here. Log KSA equals log Km minus log of the specific surface area. If we plot log of KSA versus log of Km, we get these diagonal lines here. They all have a slope of 1, and they have an intercept that's determined by the specific surface area. Keep in mind that there's some assumptions built into this model, in particular that the order of reaction with respect to the surface is 1, and that the reactive site density is in fact proportional to the bulk surface area measurement, which is usually done by BET. So this figure here, I invented this for our group for this particular purpose, but I want to emphasize that it's not completely new. There are other versions of this figure out there. Recently I found this one in the biochemistry literature, which is the same kind of graph, but it's now for enzyme kinetics. But we went on to use this very effectively in a number of cases, and I want to illustrate the most important one here. So the question was, is nano ZVI more reactive than micron-sized ZVI? And in the first few years of nano ZVI, everybody said, nano ZVI is much more reactive than micron-sized ZVI. And I found that point to be a little bit bothersome because it wasn't clear to me whether that was just because the particles were smaller and therefore had higher surface area. So we're going to use these graphs to test that question. So first look at this cartoon on the left here. Start at this position here, just an arbitrary point on the graph. and say, and then look at the arrow that I've drawn here that goes with with number 1. So in this particular case, you have a situation where you have one material with a constant type of surface site. So the intrinsic reactivity of the surface site, KSA, is a constant. So it's flat here, but as the particles get smaller, the specific surface area goes up, and you go from contour to contour, and therefore you move this way on the graph. So that's the only effect there is particle size and increased surface area. There's no change in the intrinsic reactivity. Alternatively, look at this case here, number 2. Starting here, if you have a bunch of particles that have the same surface area, but they have different intrinsic reactivities, say they're zinc and iron and magnesium or whatever, then you would track along a line like this. And those two cases are distinctly different. If you have a situation where you have the particle size getting smaller to the point that it kicks in a true nano-size effect, so the intrinsic reactivity of the surface sites gets larger, the data should go like this, and then they should move into the space that I labeled number 3. And unless you can prove that, you haven't shown that the intrinsic reactivity of the nanoparticles is higher. I've never seen a data set that actually shows that. But we can see over here, in fact, that the data that we have pretty much doesn't show that. So what we did is we compiled experimental measurements for carbon tetrachloride for the for the green symbols are for micron-sized high purity ZVI. The blue symbols are for scrap iron, so low purity micron or larger size ZVI, and the red symbols are for nano ZVI, which is generally high purity. And what you should see here is that the red points here consistently lie higher on this KM axis than the green points. So that means that on a mass normalized basis, NZVI is more reactive than micron size ZVI. But if you look over here on this axis, you see that the nano ZVI is actually a little lower than most of the micron size ZVI. So the conclusion is no, there's no evidence in all these data that nano ZVI has a higher intrinsic reactivity of the surface than micron size ZVI. In fact, it looks like the reverse. Okay, so that's 3 examples, dechlorination pathways, kinetic model, surface reactions, and 1 example of graphical methods where to illustrate my theme of ZVI serving as a model system for doing bigger and more important things than just studying ZVI. So now I'm going to change gears and switch to sulfidation. And these two things are nicely connected because I'm going to use some of these tools, these graphical methods that I feature down here. I'm going to use them as we proceed and talk about sulfidation. So I showed you already sulfidation is on this Venn diagram here. It's a significant new area of ZVI application. It's attracted a lot of attention recently. And we wrote a review paper that was published in ES&T in 2017, which is shown here on the right. And I will use that as a kind of a roadmap for the next few slides as I talk about what I want to say about sulfidation. So in that review paper, I spent a long time trying to organize the early history of sulfur plus iron literature. And there's more old work on this than most people realize. So I'm summarizing some of it here. We reviewed in the review. We started back in the 1990s, mostly with work by Kim Hayes, who was the professor at Michigan, and Liz Butler, who was his PhD student. And they did a series of really good papers on dehalogenation of chlorinated solvents by iron sulfides. No ZVI, never used the word sulfidation, but this background is very relevant. These are very high quality papers, and much of what they did applies to the new system that we're going to talk about in a moment. Starting a little bit later, there were a number of papers that combine sulfur and iron metal in a variety of ways. And so there are three of them listed here that are related to where I'm headed with this story. The first one here is from a group in Korea. Enju Kim was the student and Yoon Sok Chang was the professor. And she invented a way of synthesizing iron in the presence of sulfur that appeared to have some advantages. I'll explain that in a minute. And then I worked with them. She came to my lab for a year and we did 3 papers together. And that's this paper here. And then shortly after that, the PhD student in my lab, Damin Fan, also got on to doing studying contaminant removal with ZVI in the presence of sulfur. But none of these papers used the word sulfidation. So it wasn't quite an idea yet. We were just putting the two things together. So it wasn't until about 2015 that the word sulfidation became a word that was used in this context. And the 3 earliest papers that fit that description are this one here by Subhasis Ghoshal, who's in Ontario, this one from Damin from my lab, and another one from Wei Lian, who at the time was at Texas Tech. She's now at University of Massachusetts. And these 3 are really the first papers to use the word sulfidation in the title or the abstract. When we did the review, we thought, okay, we need a definition. And we were a little surprised to realize that nobody had really defined the word sulfidation at all, and certainly not clearly. In the early work that I just mentioned, there are only implicit definitions. Nobody actually defines the word. The earliest example of a definition is in the abstract of Damin's 2016 paper, and there we said sulfidation, for example, modification by reduced sulfur compounds. So it was a direct definition, but it was parenthetical, as in between parentheses. Only in the 2017 review did we directly define the term. So here, these are really the first explicit definitions of the term. Sulfidation refers to the reaction of metal and or metal oxide materials with sulfur species to form metal sulfides. And then in the abstract, a shorter version of the same idea, iron-based materials modified with lower valent forms of sulfur, i.e. sulfidated. So this is probably the most fair and balanced definition that you all should be using. I want to comment here just before I leave this. There's a few cases, few papers in the literature where you'll see the word spelled a little bit differently with a Z. Technically, it's not wrong, but it's not helpful to be spelling everything differently in every paper. So I would recommend that you avoid that and you spell it the way we're spelling it up here. So what is sulfidated ZVI? Well, a lot of work has been done on characterization, detailed material characterization of this stuff. And when I did the review with Damin in 2017, we invited this guy, Jan Philippe, who's in Czech Republic and has access to a very advanced materials characterization lab to help contribute to that review so that we would have state-of-the-art materials characterization in the review. In the supporting information for the review, there are some figures like the figures that you see down here, but they're only in the supporting information. And so Jan has finally got around to publishing this method in more detail in a 2020 paper that just came out. And so you can see from these images here that the particles are coated with iron sulfides, and we don't need to spend time on the details of his conclusions. The point is simply that detailed material characterization of sulfidated ZVI has been done in many papers now. So then the other question is, okay, how do you make this stuff? Well, it turns out that there are quite a variety of different ways of sulfidating ZVI. And we classified them in this table 1 that's shown over here on the left. And you notice a few general things here. For example, that on the left column, we list different sulfidating agents. That's basically the sulfur-containing treatment. we list the different types of material that's treated, and we list different types of ways of combining them. And over here, I've summarized that more fully. And I want to just mention again, the starting material matters. The type of source of sulfur you use makes a difference. But the other thing that matters is timing. So concerted versus sequential, I'm going to define in the next slide. That's quite fundamental. Another issue is exposure and aging, and I don't actually have anything on that in this talk, but that's a big deal. The last one here has to do with how you combine the materials. And I've featured in the bottom here a little bit of work by Feng He, who is, I believe, on the line here someplace. And I've worked with him a little bit recently on a couple papers, but this is his first paper on this, which I don't think is the one that I was involved in. And what he's been doing is he's been sulfidating using sulfur zero, so elemental sulfur, which is a solid. And so that's interesting because this is a solid reaction that he's doing. And notice if you look over here, this one here, elemental sulfur with MZVI, solid interaction. That's the only solid case for making sulfidated ZVI. So there are lots of different ways to make sulfidated ZVI. You get different results depending on how you do it. Now, one of the biggest factors is the timing of how you assemble the ingredients. And I want to emphasize this a little bit because it's important to the next couple slides that I want to show you. So there are two ways that this was done in my lab in the early near the beginning. First one came from the woman that I already mentioned, the student from Korea, Enju Kim. And what she did is she mixed dissolved iron with a sulfur-based reducing agent at the same time that she added sodium borohydride as a reducing agent. So she precipitated iron sulfide and iron metal together all in one pot. It was a concerted synthesis method. It produces a complicated but interesting material that, as I'll show you, has desirable properties. I don't have a picture of her. She managed to avoid getting photographed when she was in my lab, but I want to show you a little bit of her data. So the figure on the left here is a KSA versus KM plot. This is probably the second one that we published in the literature. The first one was 2011. This one was 2013. And it serves the purpose beautifully. So the question that I had for her was whether her material, which is here represented by NFEFES, so that's sulfidated nano ZVI in her old terminology, is it really more reactive than the non-sulfidated material? Well, look at the KSA-KM plot. All the non-sulfidated materials are plotted here, and the sulfidated materials are plotted here. They're higher on the KM axis, but they're also higher on the KSA axis. So I was convinced her material was actually more reactive than the unsulfidated material. But not by much, right? Just a little bit, but clearly they were more reactive. More recently, for the review that Demin Fan was the lead author on, I had a postdoc who was working in my lab just around that time who compiled additional data in order to make this new updated version of this graph here. So this was TCE. This figure over here is also TCE. This was 2013. This is 2017. These data right here, I think are these data right here, the green ones. And the open circles that are transparent, are kind of lightly colored, those are non-sulfidated materials. And the darker squares and triangles here are sulfidated materials. So you can see that actually with all these many more data from different papers, different students, different countries, that you still end up with the same conclusion. Sulfidated ZVI is a little bit more reactive than non-sulfinated ZVI, but not by a whole lot. We'd also made the figure for carbon tetrachloride. I'm not going to show that to you, but the figure for carbon tetrachloride actually shows that sulfidation does not make the reaction go any faster. And so it's important to keep that in perspective. So sulfidation may make contaminant degradation go faster, but not necessarily. All right. So now the second case down here is the one that was done by my PhD student, Demin Fan. So I have a picture of him when he graduated, unlike Inju. And in Demin's case, what he did is he synthesized nano ZVI, and then he added the sulfur. So it was a two-step synthesis process. And he also produced a fairly complex material, which we did extensive characterization. But the material is very different than the one that Inju made. In Demin's case, the original project that he worked on was very different. He wasn't studying chlorinated solvents. He was studying technetium, so metal oxyanion. This TCO4 minus is protectanate is the technical name for it. And we knew that technetium, protectanate, could be reduced to technetium oxide here, this TCO2, and that would immobilize the technetium oxide, and that would be a way to remediate groundwater that was contaminated with technetium. But we also knew that TCO2 could be reoxidized to protectanate and remobilized. The hypothesis of Demin's project was that if instead of reducing it with say iron 2, we reduced it with a sulfur species, then we might instead form technetium sulfide instead of technetium oxide. And we knew that was harder to reoxidize than TCO2. So that was the hypothesis of the project. And Demin did a detailed study where he characterized iron 0 sulfidation to make FES, FES reacting with protectanate to produce technetium sulfide, and then the kinetics of reoxidation of technetium sulfide to protectanate. It was two long detailed studies in the S&T, but the only reason that this really belongs in this talk is because in retrospect, he was doing sulfidation. We didn't call it that. We didn't really think of it as that at the time, but in retrospect, it was sulfidation. So Demin had two ES&T papers, and he needed a couple more to graduate. And so he switched gears a little bit to chlorinated solvents. And so this here is from his third major paper. And in that particular study, he started with TCE, and he used nano ZVI that was synthesized in the presence of carboxymethylcellulose. So this is so-called CMC-NZVI. He used the recipe that was developed by Feng He when he was a graduate student at Auburn University. And he got data that looked like this. So TCE is rapidly degraded, but then it stopped. And in that particular case, we measured the formation of hydrogen in that reactor, and we realized that we were forming so much hydrogen that it proved that all of the zero-valent iron was reacting with water in that bottle. And so the reason that this plateaus out is not because the CMC NZVI wasn't reactive with TCE. It was because there was no longer any NZVI there. It was all used up in the reaction with water. If he sulfidated the CMC NZVI, he got steady dechlorination of TCE all the way down to low values. And this was the real crux. He got almost no hydrogen formation. Compare this here to this here. And we realized that was a hugely important result. And so Demin summarized that in the graphical abstract for this ES&T paper with this figure here, which represents in terms of electron efficiency, where on the left case, we have most of the electrons going to reducing water and few of them going to reducing the chlorinated solvent TCE. And over here, we have the reverse. With sulfidated ZVI, we have very little reduction of water and most of the electrons going to TCE. That's a huge, important result. And I'll show you why in just a moment. But I wanted to say that because we're quite interested in that, we have developed that further. And just recently, I published a paper, again, with Feng He, who did a lot of the work in putting this together. that's all about how you quantify and characterize the efficiency term. And so there's a figure in the paper that shows that you see here on the right, and it classifies all the different branches. The electrons go this way, the electrons go this way, the electrons go this way, they go this way. Each one is an efficiency. And we wrote about how you quantify these efficiencies and how they're affected by various factors, including sulfidation. And to show that graphically, we made this plot where we summarized data from the literature, and we calculated efficiency. This is electron efficiency for all of these different experiments. So in this x-axis here, it says datum. Basically, that means a data point. So they could be an individual experiment in one paper, or they could be from different papers. Doesn't matter. Each one is a separate data point. And what we did is we color-coded them by the different types of reactant phase, and then we sorted them by their efficiency. So low down here is bad. That's where the efficiency is low, and most of the reaction is going to reducing water. And up here, large is better. That's where the efficiency is high, and most of the electrons are going to reducing the contaminant. Now, the big picture that you're supposed to see here is that these three here, blue and green, are not sulfidated. Yellow and orange are sulfidated. You see green, blue, blue, green, green. Most of the blues and greens are down on the low end, and most of the reds and oranges are up on the high end. So this proves comprehensively that overall, sulfidation is producing a more efficient treatment process, in the sense that its electrons are going to reacting with the contaminant instead of reacting with the water. So I said that this was really important from a practical point of view, and I want to show you why. So these figures come from a different, slightly older set of literature papers of ours. It's 2013. We did a pilot study where we did an injection of NZVI into a tank that was designed to measure how far the NZVI traveled in the ground. And at the time, that was the main question. What we were really mostly interested in is how far does the NZVI go? So if we have an injection well here, and you inject NZVI, then the NZVI is going to go somewhere. So that's the blue part here. And the question is, how far down gradient does it go? And what other things can you measure in sample wells to monitor the progress of the injection? And so we designed this study, and we did this study in a swimming pool-sized model aquifer. And the photograph is shown here, where we have injection ports here in the back. We have pumping, say, extraction wells here. And then these little small tubes here are sample ports. Those correspond to those sample wells here along this cross section. And we injected the NZVI here, and then we measured everything that came out down here. And we measured lots of things. Notice that the scale of this is not really all that large. So here's the injection point in the center. The first sample well is only a quarter meter away. That's probably right about here. The second one is a half meter away. That's about here. This one is a meter away. That's probably about here, and et cetera. So it's a pretty small scale of transport. We made our own CMC stabilized NZVI. We did the injection, and we made the measurements. There's a lot of data, but these pictures really tell the most important part of the story. So here you see just bottles containing the water that we took from the extraction wells. And the first set here is from well R. That's the one, that's R stands for red. That's the red point here. That's the one that's closest to the injection well. And so it's only 0.25 meters away. This one down here, well orange, is a half meter away, and this one down here, well yellow, is 1 meter away. This is time here of the injection experiment. And so what you see is that at the beginning, what comes out of the well is just water. It's unaffected by the injection. It's just the water that was already in the ground. Over time, a short amount of time, 10 minutes, you get breakthrough and it turns completely black. So you get breakthrough of NZVI, unoxidized NZVI. A little bit further away at half a meter, it looks like this. So water, water, water takes longer to break through. Eventually it breaks through as black, still reduced NZVI. A meter away, you see this. You see water, plain water, plain water, and then gradually you get breakthrough, and you get a little bit of black here way out after at 100 minutes. So, of course, originally we thought the most important result here was the breakthrough of the reduced NZVI. That's the black part, right? And so this shows that the NZVI really didn't make it much more than a meter. That was really the limit, which is not very far. But it turns out there was a much more important thing going on in this data, and that's the yellow, not the black, but the yellow. And the yellow is oxidized NZVI. How was it oxidized? It reacted with water and mineral material, mineral grains that was in the aquifer that we were doing the experiment in. So that stuff was reacting without reacting with contaminants. That's the inefficient reaction that we want to try to avoid. This shows you that by the time you get to 1 meter, all the ZVI is oxidized. So it doesn't last, it doesn't have enough capacity to be useful at this distance. Sulfidation should help a lot with this. And that's why that sulfidation effect is so important. Okay, so just I'm getting close to the end. Just a couple more slides on assorted topics. So to end up sulfidation, let me say that sulfidation is very popular with the practicing engineering community. It's already being advocated or used in technologies that are widely used by consulting companies already. There are people that are patenting it for commercialization. And then there's even one company that most people have never heard of that has trademarked and sold a product called SMI for sulfur modified iron. And they've been selling this for 25 years. And so it's clear that sulfur modified ZVI is a material that is very attractive to the practical engineering community. And so there's no doubt it's going to become quite a common thing in real world engineering applications. So now to wrap this up, I want to put this in a slightly broader context. So I've been talking about ZVI. So here I have ZVI in red, here, 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 here. So what this is this is a list of technologies that use ZVI. So SZTI stands for source zone targeted injection using NZVI. ISSM stands for in situ soil mixing, where they combine clay and ZVI and they mix it with a giant auger. That's another technology that uses ZVI. This one here called ISCR, ISCR, uses ZVI combined with complex carbon to feed microbial in situ processes. It's a trademarked and commercial technology. And then, of course, we have traditional permeable reactive barriers that use ZVI. So those things are based on zero valent iron, but there are a variety of very closely related technologies that don't actually have ZVI in them, but really belong in the same family. And I've listed some more here: in situ redox manipulation, where dithionite is added, but no ZVI. Catalytic reductive dechlorination, where hydrogen is added and a catalyst, but no ZVI. In situ biogeochemical transformation, this is another one where sulfur is added, and abiotic natural attenuation, where no ZVI is added. These things are all abiotic pathways for degrading contaminants, for treating contaminants, but they are but only some of them are ZVI based. And so together, you can group them all together into a big family, which we call in situ chemical reduction, ISCR. And I wrote a chapter on ISCR, which is cited over here on the right, in 2014. And for that chapter, I did a figure, which is kind of a map of ISCR technologies. And so here we have treatment volume, so big plumes and source zones. And here we have natural reductants that are weaker and engineered reductants that are stronger. So ZVI is over here on the strong engineered reductants, NZVI, ZVI, and then hydrogen palladium catalyst. This one is ZVI. Sorry. This one is also ZVI, but these are the ones that are that do not involve ZVI. So weaker reductants, bigger plumes. The whole family of in situ chemical reduction is becoming a thing with the consulting engineering community. And a couple years ago, the engineering company Regenesis asked me to do a webinar about in situ chemical reduction, which I did, and they recorded that, and they made it available on YouTube. And it's quite useful for people that want a systematic introduction to in situ chemical reduction, because it's a pretty comprehensive webinar. Of course, you guys can't get YouTube still, I don't think. And so last year when I was in China, I asked the people at Regenesis if they could do something to help with that. And they said, sure. They made a copy of this webinar and they put it on Dropbox. It's a private link, so you need the link. And so this QR code should take you to that file on Dropbox. So you can snap it now, or somebody can send it to you later. You should be able to access a video of my presentation of this webinar about in situ chemical reduction. Okay, very last slide here. Remember, I started talking about 25 years of ZVI, right? And it turns out, since that's a landmark date, we're going to have a party. And we were planning to have the party last year, but the party got postponed because of COVID. We're hoping to have it this year, in the middle of next year, June, end of June, beginning of July 2021. It's in Portland, Oregon, my hometown. And we have invited all of the early, kind of most important original people working in the field are scheduled to be there. So with a little, if COVID will cooperate and we're able to get everybody together at that event, it will be another great landmark in the history of ZVI, and hopefully some of you will be able to come and join us. So with that, I will stop at my final backup slide.